We're going to move on today by looking at the eukaryotes. And we'll start with the simplest eukaryotes, the Protista. What you're seeing here, by the way, are single-celled eukaryotic organisms known as diatoms, and they're surrounded by this silica shell. They build their own shell out of silica, which is glass, basically. These really are quite beautiful and remarkable organisms. Recall that we have three major domains of life. We've had a look at two of them now, the bacteria and the archaea. Hey, we're 2/3 of the way through. No, there's a lot of diversity within the eukarya. So within our domains, the next group, the next subgroup would be phylums, or phyla, if you want to use the correct plural. And we have more than 30 major phyla within the eukarya. Looking at the eukarya on the right, we have our multicellular organisms. So that's that red bubble. Everything outside of that is considered a member of the kingdom Protista. Basically, we understand what an animal is. We understand and agree what a fungus is. We agree what a plant is. Anything that doesn't fit into one of those three nice multicellular groups is thrown into this Protista. So everything else in yellow, that's Protista because, to be honest, biologists are not quite sure what to do with those organisms. They're still trying to sort out their relationships. Back to the very beginning of life, a little more than 4.5 billion years ago, we had competition among the early self-replicating molecules and then the earliest things that were almost life, proteolife, it's referred to sometimes. But something came forward as a clear winner. We believe that we can trace all of life back to one common ancestor. And this common ancestor gave rise to, again, the three domains, the archaea, the bacteria, and the eukaryota, the eukaryotes. Things were chaotic in the beginning, no doubt, but the last universal common ancestor that gave rise to all things living today is referred to affectionately as Luca. Now, why do we think that there was one universal common ancestor? Well, all living cells share some fundamental properties. The genetic code, so how nucleotides are used to code for proteins, that is universal. So it makes sense that that's something that popped up as a very primitive trait in the first living things. There are a number of metabolic pathways, glycolysis for instance. We won't get into that in detail, but glycolysis is the process whereby sugar, glucose, is split to liberate energy. And that's the basis of metabolism for all living things. So there are a number of traits, structures, biochemical pathways, etc., that link all of life back to one ancestor. That ancestor, Luca, gave rise to our three groups, the bacteria, the archaea, and the eukaryotes. And there was transfers between these groups, transfers of information, but also bacteria became incorporated into eukaryotes as organelles. We have mitochondria. Mitochondria, it's thought, came from bacteria that are very similar to E. coli that we have around today. They became incorporated, enslaved perhaps, into cells, into eukaryotic cells, and they're still there today. We'll talk about that in a moment. Plastids. Now, what do we mean by plastids? We're talking about chloroplasts here. These are organelles that harness the energy from the sun to make food for algae and plants. They can also be modified into storage structures, but those chloroplasts and the storage structures that were derived from chloroplasts are referred to as plastids. If we go back to the very beginnings of life, to the roots of the tree of life, it's really a web. There's information, there's bits and pieces being swapped back and forth between major groups, and this does still occur actually within the prokaryotes, bacteria and archaea. They swap information all the time, not something that happens in eukaryotes so much. But there was a long period of time where species were not really distinct, and these populations of cells were not really separate units. They were exchanging stuff. They were developing and evolving together. But we do have very clear evidence that two major organelles, the chloroplasts and the mitochondria that we see in eukaryotes, are actually entire prokaryotic cells that became incorporated. Prokaryotes don't have a nucleus. Prokaryote literally means before the nucleus, and eukaryotes, EU means true, true nucleus. They have a nucleus. They have a compartment. It's almost like a building inside the cell. It's a library to store genetic information. Eukaryotes also have lots of other compartments. Those are referred to as organelles. Organelle literally means little organ. Think about yourself. You have a stomach, for instance, which is a compartment that does something special. It digests your food. It's filled with acid. It's filled with hydrochloric acid. In cells, there are little compartments known as lysosomes that are filled with digestive enzymes and acid, and they break down food in the cell. Likewise, we have the Golgi body, we have the rough and smooth endoplasmic reticulum. They're all compartments that do something special. They have a specialized function. What you're seeing here are two types of eukaryotic cells. On the left, we have a plant cell. You can see there's a big vacuole, that clear thing in the middle, it stores water. We have the nucleus in purple, of course. We have endoplasmic reticulum around that in blue. The green structures are chloroplasts, which will trap energy from the sun. And then that little orange hot dog-like structure is a mitochondria, which will break down the food that this cell made using energy from the sun. On the right, we have an animal cell. We've got a flagellum there, that whip-like structure that can move it around. We don't always have those in all cells. We have these little things poking out in the front, known as microvilli. Don't have those in all animal cells, but you have them in the cells lining the inside of your intestine, for instance. Inside, we have the same stuff we saw in the plant cell. We have the nucleus in purple. We have endoplasmic reticulum in blue. That big wobbly thing in front of that is the Golgi body. And again, we have the mitochondria. So we have all of these complex structures in eukaryotes. And we'll talk briefly about how these appeared. We're going to focus on the mitochondria and the chloroplast. Mitochondria and chloroplasts are a product of something known as endosymbiosis. Endo means inside. Symbiosis means a relationship, a close relationship between two organisms. So let's look at that symbiosis part first. Parasitism. That's an example of a close relationship between two organisms. It's a relationship where one benefits and one is harmed. So imagine you have a tapeworm living inside you. The tapeworm is benefiting, it's getting pre-digested food, but it's stealing from you and you are being harmed. A more extreme example would be predation. Think of a fox and a rabbit. They have a very close relationship. The fox eats the rabbit. The fox, of course, is benefiting. The rabbit is getting the short end of the stick there. But that is a close relationship. That is a symbiosis. Mutualism is where both organisms benefit. And you're seeing a really good example of that here. So on the left, we have some termites. And termites eat wood. Now, wood is made up of the cell walls of plant cells. And wood is made up of cellulose. And cellulose is really tough to digest. You could go out into the forest and chew on a tree and you would get nothing out of it. You cannot break down cellulose. And the termite cannot break down cellulose either, not on its own. In its gut, it has a little tiny microscopic single-celled eukaryote known as trichonympha. You're seeing that on the right. This thing can break down cellulose. Cellulose is made of sugar, it's made of glucose. So termites will take in that cellulose, chew it all up, break it down into smaller pieces, and trichonympha will break it down into sugar, use some of that itself, and then pass some of it on to the termite, and everybody's happy. In fact, if you want to be really, really mean to a termite, when it hatches out of the egg, the first thing it does is it eats some of its parent's poop. And if it doesn't do that, it doesn't pick up the trichonympha and it starves to death. So again, if you want to be mean to a termite, you can take a little baby termite and hatch it outside of the colony, and it will be a perfectly healthy termite, but it will starve unless you feed it some termite poop. Okay, but the story doesn't end there. It gets even weirder. Inside trichonympha are bacteria that are living inside that cell. Remember, prokaryotes usually are a lot smaller than eukaryotic cells. So it turns out trichonympha can't break down cellulose either. It takes in the cellulose, it passes it to the bacteria that it has inside of it. That bacteria breaks it down into sugar. And that sugar is released into the gut of the termite, and the termite gets its share as well. So we have 3 organisms here, all living together, all benefiting. So that last example was a nice happy one. We have an example of endosymbiosis where we have one cell living inside another one, and both cells are benefiting. The bacteria is getting food in a nice safe place to live, and of course the trichonympha is getting food from the bacteria. So everybody's happy. But we can have situations of endosymbiosis where we have a cell living inside another cell and parasitizing it. One example would be the bacterium that causes tuberculosis. If you have tuberculosis, this is a bacterium that's living in your lungs, your white blood cells will try to destroy that bacteria. They will gobble it up and they'll try to break it down, but they can't. These particular bacteria have a really tough cell wall, and the bacteria will reproduce and live inside the white blood cell and eventually kill it. Here's another example on the screen here. We are looking at epithelial cells. Those are cells that line the inside of an organ, for instance. These are lining the inside of the urinary and genital tract, so the urogenital tract. Inside these epithelial cells, you can see some dots, and those dots are chlamydia cells. These are the smallest known cells. They're very, very simple bacteria. Chlamydia is not helping these cells, of course. It's reproducing inside the cells almost like a virus. It's such a simple cell that it doesn't even have much in the way of metabolism. Instead, it steals energy from the host cell. And then eventually the host cell will burst. It'll fill up with these chlamydia bacteria. They'll burst out and they'll go on and infect another cell. So once again, this is also endosymbiosis, but chlamydia is definitely benefiting, and the epithelial cells are definitely not. Okay, so we've seen how one cell can live inside another cell. Let's use that to figure out how eukaryotes evolved. Back to the very early Earth, we had archean cells appearing and bacteria cells appearing almost right afterwards, but there was a long stretch of time before the eukaryotes appeared. And it would appear that the eukaryotes are a mixture of bacteria and archaea. Specifically, it's thought that an archean cell took up bacteria, and those bacteria became the mitochondria and the chloroplasts. So looking at the diagram at the bottom there, we have our ancestral prokaryote, which was almost definitely an archean cell. We don't have any internal membranes. We don't have a Golgi. We don't have an endoplasmic reticulum. We don't have a nucleus, of course. But you can imagine how those could appear fairly easily. If you just take the plasma membrane and push it in, all of a sudden you have internal membranes. And if we look at the nucleus, if we look at the membrane around the nucleus, it's actually not just one membrane, it's a pair of membranes. It's kind of a pocket. So this makes sense. The plasma membrane got pushed in and it formed all of the organelles, with the exception of the mitochondria and chloroplasts that we see in today's cells. Now we have a cell with a nuclear envelope surrounding the DNA. We have an endoplasmic reticulum, etc. The next thing that is thought to have happened is that some of these cells, these modified archean cells, took up aerobic heterotrophic bacteria. Now, what does that mean? Aerobic means they like oxygen. So by the time the eukaryotes appeared, oxygen was starting to become a thing in the atmosphere because bacteria invented photosynthesis. We have these bacteria being taken in that like oxygen. They can use oxygen to break down their food completely. Heterotrophic means that they eat stuff. They're not making their food from photosynthesis. Instead, they are eating stuff and breaking it down. Now, how did this happen? No one's sure of that, of course. It could be a case where an aerobic bacterium was parasitizing a cell and became kind of trapped in the cell. It could be a case where we have our archean cell trying to eat a bacterium and not being able to break it down, much like bacteria not being able to break down tuberculosis. But for whatever reason, the bacteria that got taken into the cell became incorporated into the cell. And over time, they lost most of their DNA, they lost the ability to function on their own, and they became an integral part of the cell. Now, a little bit later on, in some of these early eukaryotes, they took in photosynthetic prokaryotes, photosynthetic bacteria like cyanobacteria. And these, once again, became incorporated into the cell, and they gave rise to chloroplasts. Chloroplasts also gave rise to something called plastids. Plastids are just modified chloroplasts that are generally used to store stuff, to store starch, for instance. The endosymbiotic theory was first proposed back in the 1970s by Lynn Margulis, a really interesting researcher with some really wild ideas, and this is one that stuck because there was lots of evidence to back it up. You might have heard of her husband as well, Carl Sagan. He was a very famous scientist in his own right, also a science fiction writer, so a really interesting couple. But anyway, getting off track here. What Lynn Margulis proposed, again, is that bacteria were engulfed by archaea, and that gave rise to eukaryotes. Now, what was she basing this on? She had some strong evidence. It was structural evidence. If we look at the mitochondria in our cells, so almost all of our cells contain mitochondria. The mitochondria have two membranes to them. They have an inner membrane that is folded and kind of wrinkled, and that membrane resembles what we see in modern day bacteria. It has similar proteins embedded in it, for instance. Around that inner membrane, we have an outer membrane, and the outer membrane is very similar to the plasma membrane of our cells, and very similar to other membranes we have inside the cell. Now that makes sense. Imagine if we have one cell engulfing another. What's going to happen is the host cell is going to reach out and grab that cell and wrap membrane around it and form a little packet, a little vesicle. So it would make sense that we would have two membranes in this bacterium that became incorporated, and that the two membranes would be quite different. If we look inside the mitochondria, we have transfer RNAs, and those transfer RNAs are different from the transfer RNAs we see in the rest of the cell. And once again, the transfer RNAs that are in mitochondria resemble what we have in bacteria. There are ribosomes inside mitochondria. They have their very own ribosomes, and those ribosomes, once again, resemble what we see in bacteria. Now, Lynn Margulis hypothesized that if she was correct with this theory, then we should also find DNA inside the mitochondria. Back in the 70s, we didn't have the technology to isolate DNA from mitochondria, but it didn't take long, and she was proven correct. Inside every single mitochondrion, there is a circle of DNA. Now that's significant because remember, bacteria have their DNA in a circle, which is very different from how our DNA is structured in the nucleus. Let's take a closer look at those membranes. So again, the innermost membrane resembles what we see in bacteria. And this membrane has been thrown into all these folds and wrinkles to increase the surface area. This membrane contains a lot of proteins, a lot of very interesting proteins. One of the proteins that's found in that inner membrane is known as ATP synthase. It synthesizes ATP, which is the molecule that's used to shuttle energy around the cell. What does the mitochondria actually do? It is the powerhouse of the cell. It does make most of the ATP, most of the energy that your cells require. The way it does that is it uses energy trapped in your food to pump hydrogen protons into the space between the two membranes, into that intermembrane space. It builds up this shell of hydrogen protons, and the entire mitochondria basically becomes a battery. ATP synthase is the only place that those hydrogen protons can move back down into the middle of the mitochondria. The middle is known as the matrix. So here we can see hydrogen protons coming down through a channel into this complex of proteins, this massive structure, relatively speaking, that is essentially an electric motor. So protons come in. As they come in, they push away protons that are already attached to that rotor, and they cause this protein machine to rotate. The protons eventually, after they've been pushed around once, they leave this molecule and they end up in the matrix of the cell. But this causes this electric motor to spin, and as it does so, it grabs onto ADP and phosphate and slams them together and makes ATP, which can then be used by other reactions that require energy. So it's a very remarkable electric motor. In fact, people that are into nanotechnology have been studying this intently because it is the smallest electric motor that we know of. It's also found at the base of flagella, but only in bacteria. So bacteria use these motors for all sorts of things. They use them to generate energy, to generate ATP. They use them to spin flagella. The flagella in bacteria spin like little propellers. And again, we don't see these special electric motors anywhere else in our cells, only in the inner membrane of the mitochondria. I mentioned that mitochondria have their own DNA, and personally I think this is probably the best evidence that mitochondria came from bacterial cells. There are 37 genes that are just found within the mitochondria, and they're found in this big circle of DNA. There are some other genes that are found within the nucleus that appear to be bacterial genes that probably got transferred from the mitochondria as it evolved to the nucleus. But definitely within the mitochondria, we have some genes that are very, very clearly bacterial, and they're used to build the ribosomes that are only found in mitochondria. They're used to make those transfer RNAs, etc. And they're also essential proteins that are needed in the breakdown of food particles to create energy. So you can't get by without these genes. If you don't have these genes, you're not going to survive. Now, one thing I find kind of interesting, and this isn't the case for all animals, but for most mammals, the mitochondria come only from the mother. So the cell that your mother produced, the gamete cell, the egg, is very, very large and contains a lot of cytoplasm. The sperm cell that your father contributed, much, much smaller, not much cytoplasm. All of the cytoplasm of the zygote, the diploid cell that would eventually become you, all of that cytoplasm came from your mother's egg. And of course, the cytoplasm contains the mitochondria. So you have your mother to thank for those 37 essential genes that are found in every single one of your mitochondria. Prokaryotes and the mitochondria and chloroplasts they gave rise to really do make the world go round with respect to life. Life would not be possible without these organisms. Life would not be possible without two fundamental processes that these organisms invented: photosynthesis and cellular respiration. Photosynthesis takes carbon dioxide out of the air, or carbon dioxide that's dissolved in water, takes that carbon dioxide, pulls the carbon off, and uses that to build food, build organic molecules. Cellular respiration, which occurs in aerobic organisms, including organisms like us that have mitochondria, takes that food and burns it in the presence of oxygen to release energy, and at the same time make carbon dioxide and water, and this cycle continues. Alrighty, let's have a look at the classification of eukaryotes. We have four big groups, four kingdoms within the eukarya. First of all, we have the fungi. We can see those here. We have the animalia. And we have the plantae, the plants, over here. And these are all fairly well understood, characterized groups. Biologists agree what makes a fungi, what makes an animal, what makes a plant. And then we have everything else shown in yellow here. These are all the other groups that have been pooled together to create the Protista. So basically, Protista consists of organisms that don't fit nicely into fungi, animals, or plantae, even though they might share some characteristics with them. Most of the protists are single-celled. Not all. There are some multicellular seaweeds, but the majority are single-celled. One useful character for setting up different kingdoms of life and for distinguishing between different sets of protists is how sex occurs within that group. Where does meiosis occur? So what's the life cycle? So remember that the very first eukaryotes were probably haploid. They had one set of chromosomes. and we show that with just an N. Occasionally, though, they would come together, they would fuse and create a diploid individual, so two sets of chromosomes, and that individual would immediately undergo meiosis to produce 4 haploid individuals. So these are single-celled organisms here, but what this event has done is it's resulted now in 4 individual organisms, individual cells that are all slightly different. They all received slightly different combinations of the genes from those two parents. And this proved to be very useful. Having variation within your offspring is a good thing because some of them might be better suited for certain tasks and certain environments. Most protists are unicellular. Again, that's not all of them. There are some that aren't, but the majority are. In animals, fungi, and plants, we have a stage of this life cycle that's become multicellular. So in animals, the diploid stage became multicellular. So just imagine that we have 2 cells coming together, that would be the gametes. They produce a diploid cell, that would be the zygote, and then instead of immediately undergoing meiosis, there's mitosis, lots and lots of mitosis, and we end up with an organism that consists of many, many, many diploid cells. That's what happened in animals. In fungi, the opposite happened. The haploid stage became multicellular, so we had mitosis in the haploid stage and not in the diploid stage. So something like a mushroom, in a mushroom cap, so the kind of mushroom that you might eat, if you flip it over, you can see gills, of course, those dark structures. Inside there, we have diploid cells that immediately undergo meiosis to produce haploid spores. But the majority of the fungus's life is spent in the haploid state. And plants, very oddly, as we'll see, they have both haploid and diploid stages that can be multicellular. Characteristic that we might use to identify the different kingdoms of the eukaryotes or to identify different groups within the Protista is how the organism feeds itself. There's 3 main ways. We have ingestion. This is where a cell or an organism takes in food in big chunks. We're talking about an organism that is a heterotroph. That means it can't make its own food. It has to get food, organic material from its environment. So we would be ingestive. We take in our food in big chunks. We have a digestive system that breaks it down, and then those little bits and pieces go in our blood, and then they travel to all the cells in our body. But we could be talking about an amoeba too. An amoeba will take in bacteria. It takes in food in relatively big chunks and breaks it down. That is ingestion. Next, we have something known as absorption. This is what fungi do. A fungus is normally living in the soil. And when you see a toadstool, like you see here, or a mushroom pop up through the soil, that is actually just the reproductive structure. But the majority of the actual organism is in the ground. And it consists of just chains of cells. Now, what these cells do is they secrete enzymes. And the enzymes are secreted into the soil, or maybe we've got a fungus that's growing on a rotten log, and the enzymes are secreted into the log. And digestion occurs outside of the cells. So proteins are broken down into amino acids, for instance. And then the cells soak that up. They absorb it. So that's a defining feature for fungi. And then finally, we have photosynthesis. This is where we have an organism that's producing its own food. So a plant or an algae cell, they will have chloroplasts, Remember, chloroplasts are derived from cyanobacteria, and what the chloroplasts will do is they'll trap energy from the sun, and they'll use that energy to take carbon dioxide apart and build food, build glucose, for instance. Algae cells and also plant cells, they will have mitochondria as well. So after they make their own food, they break it down with the mitochondria. So imagine, let's put this in a human perspective, that I put a birthday cake in front of you. Now, of course, being an animal, you are ingestive. That's one of the things that defines animals. And you would probably just reach out and grab a big chunk of the cake, or maybe you'd use a knife and fork, but you'd take big chunks off of the cake and you'd eat them. That's ingestion. But imagine if you were a weird fungal human. If you were a fungus, what you would do instead is you would reach out your hand, you'd stick it into the cake, you'd secrete enzymes from your fingertips, let's say, and break down that cake into mush and then just soak it up through your skin. That's absorption. Now, of course, if you were photosynthetic, you wouldn't care about the cake at all. You'd go outside, lie out in the sun, and make your own lunch. So eukaryotes have 3 different ways of feeding themselves. When it comes to kingdoms of eukarya, the animals are ingestive. That's one of their defining features. The fungi are absorptive, and the plants are photosynthetic. When we're talking about the leftovers, the protists, we tend to refer to ingestive protists as protozoa. You've probably heard that term before. It's a pretty old term. It's not actually a proper taxonomic group, but it's still useful. A protozoan is a protist that acts kind of like an animal. It takes in food in big chunks, for instance. It's ingestive. And then we have a group of protists known as the water molds, and another group known as the slime molds, and they're absorptive and they behave a lot like fungus. What you're seeing there is a picture of a poor goldfish, and if you've ever kept fish, you might have come across this. This is an infection with a water mold. It looks a lot like a regular mold, like a fungus, but it's not. It's a little bit different. This is actually a condition known as ick because it's icky. And then finally, we have protists that are photosynthetic, and we refer to those as algae. So protozoa, water mold, algae, these are kind of common names for groups of organisms within the protists based on how they feed themselves. Let's get into the actual groups, and don't get too hung up on the proper scientific names. I will try to use common names whenever I can. So you can see within the Protista, we have these 5 big groups. And you can see again, the actual multicellular kingdoms, fungi, animalia, and plantae, are located towards the right on our cladogram here. Note also, and we'll talk about this more, but fungi and animals are fairly closely related. In fact, you might be surprised to learn that fungi are more closely related to animals than they are to plants. Okay, we'll go through these groups here, and we'll start at the left here with a group known as the excavata. And I'll give you a couple examples of these protists. Unfortunately, these are protists that are not discussed all that often by your, general guy on the street. So we don't have common names for the excavata. 2 of the groups within the excavata are especially strange. They don't have any mitochondria. And it was thought that these groups might be the original eukaryotes. So maybe they're leftovers of a time when eukaryotes hadn't taken up bacteria as mitochondria yet. However, not that long ago, researchers have found traces of mitochondrial DNA in these cells. So instead, it appears that they lost the mitochondria. They had them and then they lost them. Now, why would they do that? Mitochondria are so useful. They allow us to extract the last little bit of energy from our food. Well, these are cells that live in areas where there's no oxygen. And when we talked about evolution, we talked about the fact that it doesn't make sense to maintain things that you don't need. It's a waste of energy. These are cells that are parasitic. So the Diplomonidida, this is a group of parasitic organisms that live in the intestine. There's no oxygen in the intestine. The Parabalsala, here we have parasitic members that live in the vagina. Again, another place you're not going to find a lot of oxygen. The Diplomonidida, they have multiple flagella sticking out all over the place. They have two separate nuclei. And the Parabalsala, they have a flagellum that's strapped to the side of the cell. It's strapped there with a thin membrane, and the flagella in eukaryotes, remember, waves side to side. So not like the spirochetes. Remember the bacteria that spin? It's different than that. We have flagella that are strapped to the side of the cell and they wave back and forth. So it looks almost like a fin on an eel or a clownfish or something like that, and it helps them get around. And here's what these guys look like. First, we have Giardia here. It causes something known as beaver fever. Not to be confused with Bieber fever, although I understand they both cause explosive diarrhea. And then on the right, we have something known as Trichomonas. This is a species that lives in the vagina, causes an unpleasant discomfort and fishy odor. A few more images of Giardia here and a costume idea if you're looking for a costume for next Halloween. Down the bottom in the right, we have a Giardia cell as it would look under a light microscope. You can see it looks like a smiley face. That's just kind of accidental. The two eyes aren't actually eyes. This is a single cell. So those are nuclei. It's got two nuclei. And then underneath that smiley face is a vacuole, a structure that's used to regulate water within the cell. It's got a whole bunch of flagella hanging off, and those would wave side to side. Above that, in the upper right, we've got an image taken with a scanning electron microscope that shows the 3D surface of these cells. We have a whole bunch of them. This is a fairly bad infection of the intestine, so they're all stuck to the lining of the intestine. A very common pathogen in many parts of the world. It's a common contaminant of water. So if you're out camping, for instance, you might want to take extra precautions and boil your water or chlorinate it with chlorine tablets, because this is something that you're somewhat likely to encounter. It causes a set of symptoms, diarrhea mainly, that's referred to as beaver fever. And the reason for that is people would quite often get sick if they were drinking water from ponds and lakes that had beaver populations. And it was thought that the beavers were the main carriers of this pathogen. It's been discovered recently, though, that actually humans are the main carrier of this pathogen, and we're the ones infecting the beavers. In the bottom left, you can see a cyst. This is one of these cells that's formed a really strong wall around itself. It's formed like a capsule around itself, and then it will be pooped out. It may end up in the water, and if you drink contaminated water, you're taking in these cysts, and they're really, really tough. They'll survive the transit through your stomach. So they will survive the stomach acid, and when they get to the intestine, they break out of their little cocoon-like structure and wreak havoc on your intestines. The last group within the excavata are the Euglenozoa, and this includes a large number of species that are free living, they're not pathogens, and then a few that are pathogens. There's a few parasitic members. So first of all, we have Euglena, and that's what you're seeing on the left bottom here. So on the left, we've got a photosynthetic cell that can, of course, make its own food. It's got two flagella. One of them doesn't even stick out beyond this pit at the back end. So there's two flagella attached in this little pit at the back of the cell. One of them sticks out quite a long ways, and it's hairy, which is weird, but that's one of the defining things for this group. We have lots of chloroplasts, which can trap energy from the sun. So that one's not going to cause you any problems because it can make its own food. The one on the right, though, Trypanosoma, this is a blood parasite. So you're seeing a sample of blood here. And of course, the round cells are red blood cells. And then those squiggly things are this parasitic Euglenozoa. It's got a very long flagellum that's strapped to the side of the cell, so the whole cell kind of waves back and forth as it swims around. This particular one causes something known as African sleeping sickness. Most members of the Euglenozoa are not parasitic, they're free living. The most common genus is something known as Euglena, and that's what you're seeing here. If you've ever seen a small pond or a puddle where the water is actually greenish, there's a good chance that you're seeing Euglena. These are microscopic little cells. They're very active. They swim around, but they are photosynthetic. They have chloroplasts, and they can make their own food. If there's enough light, they have a little eye spot, a little red dot that they use to sense light, and they move towards the light. But if you put them in the dark for a few days, they change. They become almost like amoeba, and then they start gobbling things up the way an animal would. So they swim around, they move a lot, which is something we associate with animals. Animal, the word, comes from animated, which means movement. And they are also photosynthetic at the same time, which is something we associate with plants and algae. And again, if you put them in the dark, they are ingestive. There has been a battle for centuries between zoologists, people who study animals, and botanists, people who study plants. The zoologists like to call these guys Euglenozoa. Zoa means animal. And the botanists like to call them Euglenophyta. Phyta means plant. So this is just one example of how confusing protists can be, because some of them don't act like one particular group. They have characteristics from both plants and animals, or fungi and animals, etc. They're really hard to classify. Moving on. Next, we're going to have a look at a group known as the chrome aviolata. Now, again, I'm going to try to stick with more common names if I can, names that you've heard of. But we'll start by looking at a subgroup, the aviolata. The aviolata are defined by the fact that they have little spaces, little sacs, underneath their plasma membrane. Alveoli means sac. You probably realize that you have alveoli in your lungs. Those are the little tiny microscopic sacs that you breathe into. Now, no one's entirely sure what these little sacs under the membrane do, but they appear to be involved in regulating the movement of water in and out of the cell. Anyway, they quite often will have cilia for mobility to help them move around. And we have three groups here. We're just going to touch on them very briefly. Again, the point here is just to give you a sense of the amount of diversity within the Protista. We have a group known as the dinoflagellates, which are really quite interesting. They have a protective shell. So this is a single cell, but it has a protective shell made out of something very similar to cellulose. and it's spiky to help protect it against anything that might eat it. It has grooves, and the grooves have flagella inside them, and it swims around. It looks like a little spaceship. It's photosynthetic. Then we have the apicomplexans. Those are all parasitic. Malaria is caused by a protist known as Plasmodium, and it fits into this group. All of these members are parasitic, and they have at one end of the cell devices, these little pointy little proteins, almost like knives, and they have enzymes that they can use to punch holes into host cells and damage them. So if you have malaria, that little protist, that little single-celled organism, is going to punch holes in your red blood cells, reproduce inside them and rupture them and destroy them, and also do something similar to your liver cells. And then finally we have the ciliates. Big group includes Paramecium, which you've probably heard of, and a lot of other species that are almost entirely free-living and non-parasitic. Just a quick little story about dinoflagellates before we continue on with the ciliates. Dinoflagellates can sometimes explode in numbers in the ocean, and that's what you're seeing here. You're seeing something called a red tide. There's so many dinoflagellates that they're changing the color of the water. Now, dinoflagellates are really important. They're photosynthetic, they make their own food, and they serve as food for lots of other things. They're the basis of many food chains in the oceans. They're also eaten by shellfish, so clams and oysters and mussels. And interestingly, dinoflagellates make a waste product which acts as a neurotoxin on us. You might have heard of paralytic shellfish poisoning before. That's where we have one of these massive blooms or explosions of dinoflagellates, and there's so many of them that they poison the flesh of the shellfish that we eat. So of course we have to be careful of that. Okay, let's continue on looking at the ciliates now. Here are a few examples. So the Aviolata contains this big group known as the ciliates, and they are covered in cilia. Cilia are just tiny little flagella, basically. They have the same structure. So cells that have flagella usually just have a few, but cells that have cilia, they might have hundreds or thousands covering the whole surface. So in the case of Paramecium, and you're seeing that on the bottom left here, they're covered in cilia. They use that to move around. They also have a structure called an oral groove, and they will use cilia to waft food into that groove, things like bacteria. And then they get taken into the cell, they get digested, and they actually have an area at the back of the cell that's kind of designated as the cell anus. That's where all the waste gets dumped. So these are complex cells. They're big cells. If you have good eyesight, you can just barely make these out with your naked eye. So you can put some on a slide, you can put it on a black background, and you'll see little specks moving around. They are big, complicated cells. At the top there, on the left, we have something called stentor, which is kind of like a paramecium, but it's stuck to something at one end. And it has an oral groove at the top, and it has a ring of cilia. It doesn't move around nearly as much. It doesn't swim around. Instead, it stays in one place, and it uses that ring of cilia to catch food. Next to that, the upper right, we have Vorticella. Same kind of thing. They look almost like little flowers, but they have a ring of cilia, and they use that to capture food. Down the bottom right there, you're seeing a pair of Paramecia mating. So even though we're dealing with single-celled organisms here, they do have sex. There are, of course, advantages to having sex. What's interesting in Paramecia is they have lots of nuclei. They have a great big nucleus, and then they can have several smaller nuclei. The big nuclei have multiple copies of the genetic material. It's really weird, and they can swap nuclei, which is similar to conjugation in bacteria, all around kind of odd. And here we have some examples of some ciliates. Down the bottom left here, we have a paramecium. You can see it's covered in cilia. And as it rotates, you can see there's a groove. That's the oral groove. So it'll move around, but it'll also waft food into that groove and eat it. They're sometimes described as being slipper-shaped. So they're shaped kind of like a slipper. I guess you would put your foot into that oral groove. Here's another paramecium here, and it is a very complicated cell. And it has some sensory organs. It doesn't have eyes or anything like that, of course, but it does have receptors, and it can sense food. It can smell, quote unquote, food and go after it. This here is something known as a stentor, and you're seeing the mouth end of the stentor. So it's got a ring of cilia. It creates a current that wafts in bacteria and other small things that it can eat. Those really small things you see moving around are bacteria. Oh, just grabbed a few. And then there's another large cell there, another large ciliate that has gotten trapped in the current. We've got another stentor up the top here. Again, you can see it's creating a current to try and feed. And finally, one more ciliate here with a really, really deep oral groove. You can see it's using those cilia to move around, and you can also see cilia within that oral groove, and it's trying to catch little tiny organisms in the water. The next large group we'll look at are the Stramonopila. And we're not going to focus on that term. Don't worry about that term. We're going to look at some of the large, most recognizable groups within the Stramonopila, and that would be the water molds, the diatoms, and the brown algae. Water molds are also known as Eumycota. Eu means egg, and they have these large egg cells, as you can see in the photo there. Now, Eumycota, or water molds, outwardly resemble fungi, and they're absorptive like fungi. They look like a mold, which is a fungus. However, their life cycle is different. The multicellular form, the mature reproductive form, is not haploid. It's diploid, which is different from fungi. The diatoms are single-celled organisms that create this miraculous shell. The shell is made out of silica, glass, basically, and you're seeing that on the left. And they come in a variety of different shapes and sizes. There's people that position them under a microscope and make art with them on a slide. And then we have the brown algae. The brown algae are the largest protists, physically speaking. So this group includes kelp, and kelp can be 40 meters in length. It's a very large multicellular organism. Water molds are most commonly parasitic, and they can parasitize a number of freshwater animals. Things like the poor salmon that you can see up the top there. And also amphibians can be really hard hit by outbreaks of water mold. Down the bottom right, you're seeing a water mold, and you can see that there's long chains of thin cells. This is what we see in a fungus mold as well. There are round structures, and those create eggs, and then the eggs are fertilized by sperm. But these can also reproduce asexually. And those long oval structures that you see contain spores that have been produced through mitosis and not meiosis. The spores are very weird in that they have two flagella. They swim around. They have one smooth one and one hairy one, and that's a defining characteristic of this group. You can see that in the top right. Now, in addition to parasitizing animals, they very often parasitize plants. Most notably, in terms of human history, they caused the potato famine in Ireland in the middle of the 1800s. So potatoes were the staple diet, especially of poor Irish people, and yeah, it resulted in a very, very large number of deaths. Diatoms live mainly in the ocean, although there are some freshwater species as well. They float around near the surface of the ocean and they're photosynthetic. So they take up the sun's energy and use it to make food, and they're an important part of the ocean food chain. There are so many of them in the ocean that when they die, their little glass skeletons sink to the bottom of the ocean and build up in layers that can be hundreds of meters thick. These little skeletons are fascinating. All the different species have different shapes, but keep in mind there's one cell inside, and this shell is made up of two parts glued together. You might have heard of diatomaceous earth. This is a white powder that's just made up of these glass skeletons. You can use it to help get rid of insects. You can spread it around in your house or in your garden. And what happens is the insects will breathe it in, and because it's basically glass, it will cut their insides open. It'll cut their respiratory tract apart. Not that pleasant for the insect. Also, if there's enough of these diatoms and they're put under pressure through geologic forces, they can be compressed into chalk. The White Cliffs of Dover are a very famous part of the English coastline. And here we've got hundreds of meters of diatoms that have been compressed into chalk. So that white material is just billions and billions of little glass skeletons, all of them microscopic, all of them a little bit different. Old school chalk, not the stuff that's usually used today, but the old chalk was made up of diatoms as well. So when you're writing on a chalkboard with a piece of chalk, you're leaving a trail of billions and billions of little diatom skeletons. As mentioned, brown algae are also a part of the Strammonopila. They have a life cycle that is very similar to what we see in plants. They have an alternation of generations. Now we'll talk about that a lot more when we talk about plants, but what this means is that they have a multicellular haploid stage and they have a multicellular diploid stage. So at the top there we have something called a sporophyte, and a sporophyte produces spores. It produces those spores by meiosis. So the sporophyte is diploid, it's 2N. And the spores that are produced by this seaweed are able to swim around. They have two flagella, one smooth, one hairy. They swim around and then they develop either into a female gametophyte or a male gametophyte. Now these are little tiny seaweeds. They're almost microscopic. You can just barely see them, and they're multicellular, but they're haploid. Now what happens is the female gametophyte produces an egg, and the male gametophyte produces sperm. And again, the sperm have two flagella, one hairy, one smooth, and the sperm and the egg come together and they produce a zygote, and then that develops back into our sporophyte. Don't worry too much about those stages just yet. We will come back to this because, as I mentioned, this is exactly what happens in plants. Plants also have a multicellular diploid stage and a multicellular haploid stage. And we only refer to the multicellular parts of the life cycle as generations. Here's the largest example of these seaweeds. This is the sporophyte, so this is the multicellular diploid individual, and this happens to be kelp. Now kelp is really common. You'll find it washed up along the shores of British Columbia if you walk along the coast a bit. And you can see that it has these round spheres there. Those are actually little buoyant bubbles. They're filled with gas, and they help keep this thing growing upright. This is a kelp forest here. Lots of life in a kelp forest. It does look like a forest, but these are not plants. These are protists. And again, these are the largest of the protists. They can be up to 40 meters in height. Continuing on in our quick little investigation of the diversity of Protista, we've talked about the excavata, we've talked about the chroma aviolata, and now I'll very briefly talk about the Rhizaria. And the only member of the Rhizaria that I'm going to touch on are the foraminiferans, or forams. They're single cells. They're fairly large cells though, and they create a shell that looks almost like a snail shell. And that's what you're seeing here. You're seeing a collection of different foram shells. What they do is they start out in the middle of this spiral, and as they grow, they increase the size of the shell, and they move from the middle out towards the ends of the shell. So we had a cell that started here, looking at the shell in the bottom right, and as it grew a bit, it moved into this second little apartment that it manufactured, and it closed off the original living space, and then it did it again, and then again, and then again, and then again, and then again, and we end up with this spiral shell. In some parts of the world, the oceans have so many of these forams that you can have beaches that are made up entirely of these tiny little shells. So instead of the beach being made up of grains of sand, it's made up of microscopic foram shells. The next group that we'll look at is a rather important one because it includes us. This is the amorphia, which literally means without form. It includes the fungi and the animals, and then also the groups in yellow, which are protists. They don't fit nicely into any of the other three domains of Eukarya. We have what are called the amoebozoa, and they're all amoeba-like. So they move around the way an amoeba would, or the way a white blood cell would. That's something that's interesting. In animals, we have amoeboid cells. We have the fungi and the animalia, of course, and then in between we have something called the choanoflagellates. And this, it's thought, are the organisms that gave rise to the animals. So we won't talk about them too much just yet. We'll talk about them more when we talk about the evolution of animals. I should mention that amorphia is also sometimes referred to as uniconta, which means single flagellum. And that's a feature for many members of this group, us included. We only have one cell that has a flagellum, and that's the sperm, and there's one flagellum. We see a similar thing in the fungi and in many of the other groups as well. The amoebozoa use structures known as pseudopodia to move around. A pseudopodium, that's the singular, is a lobe-like structure, and pseudopodium means false foot. So they have this lobe that they push forward, and then they push all of themselves into that lobe. That's how they move. Imagine if you could put one foot forward and then squeeze your whole body into that foot and then do it over again. This is how they get around. They also use the pseudopodia to engulf their food. So here we have a rather greedy amoeba that is gobbling up a diatom. It is indeed a rather interesting way to get around. What you're seeing here on the left is an amoeba moving in real time. It's forcing its cytoplasm into these pseudopodia. On the right, same idea. This one's just been sped up a little bit. Perhaps the most fascinating of the amoebozoa are the slime molds. This is another example of a protist that resembles a fungus in some way, and it's referred to as a mold by its common name, but it's not technically a mold. So molds are fungi. That term really should only be used when we're talking about fungi. But here we have hundreds of thousands of individual amoeboid cells that come together periodically and behave almost like a multicellular organism. They quite often have very bright colors. No doubt you may have seen some of this on a rotting log and not thought anything of it. Or you might have come across it and not even realized it was alive. This is a very common slime mold known as dog vomit fungus. Now again, it's not a fungus, but it does look a lot like dog vomit. If you saw it, it might have grossed you out. However, if you came back and looked at it the next day, you might be surprised to see that it's changed shape, or perhaps it's moved. There are two broad groups of slime molds. Let's start with the plasmodial slime molds on the left here. Notice that they spend most of their existence in a diploid state. We'll start down the bottom with a structure called a sporangium. This is a diploid structure that produces spores. The spores are produced by meiosis. So we have diploid cells that undergo meiosis. Each of those diploid cells is going to produce 4 haploid spores. If these spores land in the water, they give rise to these flagellated cells that swim around for a bit. If the spores land on land, they hatch out as amoeboid cells. Either way, these cells are going to seek each other out, and we're going to have 2 of these amoeboid cells come together and produce a zygote. Now the zygote is diploid because we had 2 haploid cells come together to make it. And what happens next is rather interesting. We have mitosis, which is asexual division. But it's just the nucleus that divides. So the nucleus divides into 2 nuclei. And now we have 2 nuclei in one cell. The cell does not divide. And then those 2 nuclei divide into 4 nuclei, again, without the cell itself dividing. And we end up with something called a plasmodium. A plasmodium is a big chunk of cytoplasm with lots of nuclei. So essentially we end up with this massive cell that contains thousands or hundreds of thousands of nuclei. And then this plasmodium starts to wander around and it starts to seek out food. And I'll show you a video of that in just a moment. Now this is different from the cellular slime molds. They don't have this plasmodium stage where they're one massive cell with lots of nuclei. Instead, there's lots of individual cells that come together and behave in a coordinated fashion. So again, let's start at the bottom here with these so-called fruiting bodies. Now these are produced asexually. This fruiting body is haploid and it produces spores by mitosis. So haploid cells can undergo mitosis and produce more haploid cells. Those haploid spores will float through the air. If they land on some damp ground somewhere, they'll hatch out as amoeba and they wander around on the ground. And all of these amoeba will seek each other out. They'll find each other and they'll come together and they'll form this big mass of individual amoeboid cells that can wander around in a very coordinated fashion. In fact, they tend to arrange themselves into what's called a slug, and it looks kind of like a slug. And you're seeing that in the photo down the bottom right. A lot of the time, they'll reproduce asexually. It's faster and easier, but we can see at the top there the short little sexual reproductive cycle. So what can happen is sometimes these cells will separate off again from the mass. They'll come together, they'll form a zygote, a diploid cell, and then that immediately undergoes meiosis and takes us back to individual amoeba, which again can wander around, pair up with other cells to have sex, or come together in a big mass and wander around and gobble things up as a massive, weird, not quite multicellular thing. Okay, here's a plasmodial slime mold in action. What we've got here is a Petri plate that has some oats sprinkled on top of it. So like the kind of oats you might have for breakfast if you're eating oatmeal. And then on the right, we have our slime mold. Now it is seeking out food, and it likes the oats. So remember, this is essentially one massive cell that has hundreds of nuclei. When it finds the food, it leaves little trails of cytoplasm between the food. So now this whole cell is very efficiently finding pathways to each of these food sources and absorbing them. And here are some videos of cellular slime molds in action. So again, cellular slime molds, individual cells, hundreds of thousands of them. We can see in this little video here them coming together. This is a time lapse. They're coming together and they're forming a fruiting body. That's the structure that they're going to use to make spores. And basically, to make the spores, all that happens is some of those amoeboid cells wall themselves off with a really tough coat, and that's hopefully going to help protect them as they're dispersed through the air. And a few more videos of the same idea here. The cells that make it to the top will become spores, and they can fly off and start a new life somewhere else. But the ones that form the stalk at the bottom, they're going to die. And one more here. Again, the spores that will be produced in those little sporangia, those little circular structures at the top, are just amoeboid cells that have walled themselves off. Now they can move around in a coordinated fashion. So what will happen is the individual amoeba cells will wander around on the forest floor, gobbling up food, and then late summer, early fall, food starts to become a bit more scarce, things get a bit colder, and then it's time for them to aggregate, to come together, wander around, find a good spot to make spores, or to actually have sex. If they're wandering around, they do it like this. So this is called a slug, not actually a slug. It's a whole bunch of individual cells that are working together somehow in a coordinated fashion. What I find really fascinating with that last example is that when we have that slug moving around, the cells are all racing to the front of the slug, and the ones that are the healthiest get to the front. And those are the ones that are going to give rise to spores, and those are the cells that will contribute to the next generation. They're fitter than the cells that can't get to the front of the slug. It really is a weird way to reproduce and disperse yourself. So imagine if humans did something like this. Imagine you lived in a small town, an alarm went off, and all of a sudden everyone in the town ran together, and then formed a big human pyramid. And the people at the top had parachutes and they floated off and they formed another town somewhere. I mean, it's just absolutely bizarre. And that leaves us with one major group. And that major group is the Archaeplastida. This includes the plants. So again, remember, we have 4 kingdoms. We have the fungi, the animals, the plants, and then everything else in yellow. That is our 4th kingdom of eukaryotes, the Protista. In the Archaeplastida, in addition to the plants, we have the Rhodophyta. These are the red algae. These are some very common seaweeds, including some you might eat. And also the Chlorophyta, which are the closest relatives of the plants. The red algae are very common seaweeds. They do look a lot like plants. They're not, though. They are protists. They're multicellular. They have an alternation of generations, so they do alternate between a large multicellular diploid individual and a small multicellular haploid individual, just like the plants do. They contain a different pigment, something known as phycobilin, which is not something found in plants. And for that reason, they have a different color. They tend to be well adapted at living in deeper water, and this pigment helps trap sunlight that's able to penetrate to that depth. Nori, which you might have eaten, especially if you're a fan of sushi, fits into this group. And finally, we have the green algae, more properly known as the Chlorophyta, because they contain chlorophyll. They also have chloroplasts that are very similar to what we see in plants. The chloroplasts in the red algae, they have a slightly different structure to them. There are unicellular Chlorophyta, so unicellular green algae that you might find in ponds, for instance, and there are multicellular forms as well. Perhaps my favorite green algae is Volvox. Now, Volvox is a rather common algae that we can find in local ponds and lakes, and it consists of a hollow ball of cells, and inside that are more hollow balls of cells. This is something that can reproduce asexually, and that's what it's doing here. So the little hollow balls inside the bigger ball are daughter colonies. This is an organism that's kind of on the cusp of true multicellularity. And we can see one of those daughter colonies breaking off right now. But they can also reproduce sexually. Alrighty, to finish off, we have our concepts. Protists are eukaryotes. And like all eukaryotes, they have nuclei in their cells. They have organelles. Although remember, some of the ones we talked about have lost their mitochondria. The Diplomonidida have lost their mitochondria because they live in areas without oxygen and they don't need them anymore. The Protista is a polyphyletic group, and the ancestry of these organisms is uncertain. So if we took all of the Protista and traced them back to one common ancestor, that would include all of the eukaryotes. Many of the Protista are animal-like, or plant-like, or fungi-like, but also they have differences. And in some cases, we have Protista that resemble more than one of those other kingdoms. This is an area of ongoing research and debate. And in fact, the names that I used for some of the groups, if you look in a different textbook, you might see different names being used. This is an area of research that's in constant flux. It's changing all the time as we learn more about these fascinating little critters. The Protista include photoautotrophs. Those are organisms that can make their own food using sunlight. Free-living heterotrophs, that's stuff like us. So organisms that feed off of other organisms in their environment without parasitizing them. Parasitic heterotrophs and mixotrophs. Mixotrophs are things that can do more than one thing. They can do a mix of stuff. Remember, troph means to eat. Mixotrophs can mix things up. So for instance, Euglena, it's an autotroph most of the time, a photoautotroph. But if you put it in the dark, it becomes a heterotroph and it starts eating smaller organisms. Most of the Protista are unicellular. Some are colonial. That means the cells come together, but they don't necessarily have separate distinguishable roles. And then a few are considered multicellular, or at least on the cusp of being multicellular. In true multicellular organisms, there are specialized cells that have specialized functions. Figuring out relationships within the Protista is very difficult, but fortunately, the analysis of DNA sequences has really been helping out quite a bit. There's a lot of work to do, though. There's a lot of these organisms that need to have their DNA sequenced. It's an ongoing issue, and certainly if we were to check back 10 years from now, there would be a slightly different tree. It's becoming more and more refined, however. Now, one of the key things that I'd like you to get out of this discussion is that the protists hold the key to determining the ancestries of plants, animals, and fungi. Plants, animals, and fungi are large groups, large, well-defined groups, but they all stem out of the Protista. I quite like this kind of tongue-in-cheek diagram here. We had the bacteria and archaea together contributing to the protists. And the protists, the first eukaryotes that appeared, tried out a whole bunch of different strategies. There's so many different life strategies within the Protista. And some of those strategies gave rise to the plants, fungi, and animals. So plants, animals, fungi, they all have their roots in the Protista. And the Protista can kind of be thought of as leftovers from that early radiation of eukaryotes. And if we want to figure out this whole mess, we have to figure out the Protista. And some terminology here. And please note that I am not throwing all of those group names at you. I've just got a few here that I'd like you to focus on.